So, in the last podcast, we talked about matter and energy, and we gave a working definition of matter as anything that occupies space and has mass. And we pointed out the fact that everything in the universe is either matter or energy. And most of the solid, liquid, or gaseous stuff that you experience in the world around you is built out of atoms. Atoms can assemble via chemical bonding to form molecules. Molecules can form macromolecules. And macromolecules, if there are enough of them together, can form the smallest unit of living matter, which is the cell. So that's what we're going to start out discussing today. So it's the smallest living unit. On average, a cell is about a millimeter in diameter, but there are quite a few exceptions. The cell theory states that cells are the building blocks of all life. All new cells come from the division of pre-existing cells, and they're the smallest unit performing all vital physiological functions. And you might think, well, of course I knew that, but for the longest time we had no idea what living stuff was composed of. It wasn't until the invention of the microscope by a gentleman named Anton von Leeuwenhoek that we were able to observe the microscopic structure of tissues. It was his experiments with cork, observing the microscopic structure of cork, that coined the term cell. He observed small air pockets inside preserved slices of cork. He termed that cells, and then he started looking at other living material, and he found that all living material that he observed had a cellular structure. And so that formed one of the fundamental building blocks of the cell theory, which is that all life is composed of cells and that all cells come from pre-existing cells. That was Schleiden and Schwann who basically fleshed out the cell theory. Cells have a wide variety in size, shape, and function. All cells in the body come from, initially, a fertilized ovum or egg. The ovum, when it's fertilized, divides into multiple cells. The daughter cells will specialize and differentiate, meaning what? Well, they'll start to look and act differently from each other as we begin to form a new human being. Now, how does this happen? Well, we're going to find out that there are chemical signals that are laid down in the proximity of the fertilized egg and within the cells that make up the fertilized egg. So they're both outside and inside these cells, that tell the cells what to do and how to do it. Essentially, what happens as cells specialize and differentiate is they begin to read from their complement of information, their DNA, in a more specialized fashion. Initially, a cell can read from any part of the DNA and use that information to direct its functions. But as a cell matures in human development, the available information becomes restricted. Okay? There are chemical ways to modify certain parts of the genetic material so that, let's say, a cell that will eventually be a muscle cell will, over time, only be able to read from the muscle cell handbook of DNA, a nerve cell from the nerve cell handbook, an epithelial cell from the epithelial cell handbook, and so on. The other information is still present in the nucleus, but it's not accessible in order to use to essentially direct the cell's activity. We talked about genes in the last installment, and genes are simply information packets carried in DNA that are protein building instructions. And if we restrict which protein building instructions that a cell can use, we're going to change its appearance, its behavior, and the kinds of things that it can produce. And that's exactly what happens in human development. Differentiation produces 4 types of body tissues: epithelial, connective, muscle, and neural tissue. And they all look and act very differently from each other. Cells are surrounded by an extracellular environment. Sometimes it's a fluid, sometimes it's a semi-solid matrix. Okay? Fluid between the tissue cells is called interstitial fluid. The fluid inside the cell is called the cytosol, and the components of the cell are held together by the cell membrane. The cell membrane forms a border around the living cell, and that keeps all the functioning parts of the cell in close physical proximity to each other. So that's one of the hidden properties of the cell membrane, is that it confines the cellular components to a specific physical space. Each cell is surrounded by this plasma membrane. The cytoplasm is found between the cell membrane and the nuclear membrane, and it has a varying consistency. Sometimes it's rather fluid, in which case we call it cytosol. There are organelles within this fluid that do different jobs for the cell. They're like little rooms in a factory that are designed to perform a particular process. Membranous organelles have a phospholipid membrane around them, while non-membranous organelles are not completely enclosed by membranes. And so here we see sort of a diagram of a cell. Okay, we have a plasma membrane surrounding the whole process. Okay, the whole living unit. Inside we have cytoplasm, which contains cytosol and organelles. The organelles can be divided into membranous and non-membranous. Examples of membranous organelles include things like mitochondria, lysosomes, Golgi apparatus, and non-membranous organelles include things like cytoskeletal elements and ribosomes. And we'll get into each of these in more detail as we go on through the rest of this particular presentation. The nucleus contains nucleoplasm, fluid, that essentially makes up the bulk of the volume of the nucleus, as well as enzymes, proteins, and very importantly, DNA, which is the information molecule that the cell uses to direct its activities. It's surrounded by a nuclear envelope, and it controls metabolism and protein synthesis. So basically it dictates what the cell is going to do, what the cell is going to look like, and the kinds of things that the cell can make. It stores and processes genetic information. Peroxisomes have enzymes that break down harmful toxins that can build up in the cell, as well as break down fatty acids and other organic compounds. Lysosomes are like the stomach of the cell. They contain digestive enzymes that break down large organic compounds, damaged organelles, and pathogens, while microvilli are membranous extensions that provide an increased surface area that aids the cell in the absorption and the secretion of materials. Golgi apparatus is a stack of flattened membranes that are designed to modify and package proteins before they're sent to their final destination in the cell. So, in a sense, the Golgi apparatus is like the distribution center of proteins for the cell. Other organelles include the endoplasmic reticulum, which is a network of membranous sheets and channels that synthesizes and transports substances. The smooth endoplasmic reticulum has no attached ribosomes and is the site of lipid and carbohydrate synthesis, while the rough endoplasmic reticulum has ribosomes bound to its surface and is the site of protein synthesis and modification. Ribosomes actually build the proteins. They are made up of a conglomeration of RNA and protein. They are sometimes bound to the rough endoplasmic reticulum. They can also be found scattered in the cytosol. They are where proteins are manufactured. Mitochondria are double membranes with the inner membrane folded in little indentations that are called cristae, and that's again to increase the surface area of the inner membrane. It produces most of the cell's ATP. Now, if you remember from the last lecture, ATP is that form of energy that's immediately available for the cell to do work. You could think of ATP as biological gas in the tank. In fact, let's write that down so we don't forget it. Okay, so we'll do this. We'll do it in red, right? So when we're talking about ATP, this is biological gas in the tank. It's a form of energy that can be immediately used. The cytoskeleton are proteins that maintain the shape of the cell and also serve as a way of moving organelles inside the cell from one place to another. It's also important in a process called mitosis, where cytoskeletal proteins act to move genetic material into two brand new cells. They strengthen and support the cell. They can also aid in some cases in the cell's movement. There are cytoskeletal elements, for instance, that aid with the ability of certain cells to move between other tissues. Examples include white blood cells and macrophages, for example. And there are also cytoskeletal elements that are important in the movement of cilia and flagella, which are appendages that allow a cell to move from one place to another, in the case of a flagella, or in the case of cilia, for a cell that's fixed in its position to move stuff across one of its surfaces. So here you can see an example of a typical animal cell. Not every cell has all these features, but many cells have many of these features, and you can see where these different elements are located, right? The nucleus with the chromatin, which is DNA plus wound around protein, okay, to compact genetic material so it's not damaged. The nucleolus, which is the site of ribosome synthesis. The nuclear membrane, the rough endoplasmic reticulum. You can see some smooth endoplasmic reticulum. They're actually contiguous organelles. And then you've got the Golgi apparatus, the distribution center for proteins in the cell, microvilli on the cell membrane, lysosomes, peroxisomes, mitochondria, okay, and also centrioles, which are where microtubules are put together and broken down. Okay, the cell can synthesize or break down microtubules as it needs them. And microtubules are part of the cytoskeleton, which is made up of intermediate filaments, microfilaments, and microtubules, and they all serve as a way to move material around inside the cell that's already there, and also to aid the cell in moving stuff across its surface, or in some cases the cell itself moving. The plasma membrane is a selectively permeable barrier that separates the inside of the cell from the extracellular fluid. It's made up of a phospholipid bilayer, two layers of phospholipids with the hydrophobic heads at the inner and outer surface and the hydrophobic tails in the interior of the membrane. So if we were to draw this, let's draw what this might look like. We'll use blue here. So if I were to do a diagram of a phospholipid, remember that the heads are polar, okay? This is just a cartoon. This is not an exact representation of the chemical structure of a phospholipid, but remember that there are long hydrophobic tails and polar heads, and so these guys form a bilayer in the cell membrane with these tails projecting towards the interior and the heads either coming into contact with the extracellular fluid or, in the case of the heads on the other side, coming into contact with the cytosol. Okay? So this is just a cartoon of what we imagine the cell membrane looks like. Okay? Now, very important that we understand here that this portion of the cell membrane is hydrophobic. Okay? And this portion of the cell membrane here is hydrophilic. This portion here, and also this portion up here. Okay. The reason that that's critical to understand is because that's part of what makes the cell membrane a semi-permeable barrier. Greasy molecules. Let's draw an example of a greasy molecule here. We'll do it in black. can very easily slide through the cell membrane from the outside to the inside. So if we imagine that this is the outside of the cell and this is some greasy molecule, he can easily move straight through the phospholipid bilayer and into the inside of the cell. This would be the inside. And the reason he can do that is because he is hydrophobic, okay? Hydrophobic. Let's put an arrow here saying he's hydrophobic, just like the interior of the membrane is hydrophobic. So there's not a big energy barrier to him sliding right through and getting inside the cell. But large polar things like proteins and salts and ions, they can't get through. Okay, so if we say here polar molecule, he has no way to get through unless he has some sort of a pump or a tunnel that's usually made out of protein that will allow him to get across this greasy middle of the cell membrane. So we can imagine here a protein that provides a passageway for this particular polar compound, okay, and then he can slide right into the hole and then go to the other side because there's a special door for him. And in some cases the door is always open in the case of what we call leak channels. In some cases the door can be opened or closed depending on cell conditions, and so that allows the cell to let certain things in and keep other things out as the cell's needs change. Integral proteins can't be removed without damaging the membrane. Some are pores or channels, as we indicated in the diagram. The glycocalyx is a superficial membrane carbohydrate layer that serves as a method of cell identification so that our immune system recognizes cells as belonging to us and therefore won't attack them. The glycocalyx also serves as a portion of the extracellular matrix, and that's important as a way of sending cells messages. It's also important as a way of stabilizing the cell's position in a tissue, okay? And it also serves as a cluster of molecules that can be recognized by other cells in the event that they have to interact in some way. It also has a lubricating function, and it can bind to extracellular structures. So this is a look at the plasma membrane in a diagram. This is a little bit better drawing than I did, but you can see the phospholipid bilayer. There are the nonpolar tails in the core and the polar heads projecting to the outside or to the inside of the cell. You can see glycocalyx components here, and then you've got these membranes with these special tunnels and gates, right? Integral proteins which span the cell membrane. They can act as a door that the cell can open or shut whenever it needs to move a particular compound into or out of the cell. And then we've also got proteins that span the cell membrane attached to cytoskeletal elements and also to extracellular matrix components, and may also happen to have sugar groups attached to them, so they're also part of the glycocalyx. So what we say with the membrane is it's what's called a fluid mosaic model, meaning that most of it is phospholipid, but you will occasionally find proteins and sugars either embedded in the membrane all the way through it, or on one side of the membrane or the other, or attached to one side of the membrane or the other in the case of these sugar molecules. So take a listen to the cell membrane barrier, this little video up here that I'll run here.
Like the walls of a house or building separate the interior from the outside world, the cell membrane is a barrier that separates the contents of the cell from the external environment. Cell membranes are a mosaic of protein and lipid molecules, both of which can drift from place to place within the membrane. Most of the surface area of the membrane consists of a type of lipid called phospholipid. Each phospholipid molecule has a polar head and a nonpolar tail. The phosphate-containing head is hydrophilic or water-loving because its polar structure is attracted to water molecules, which are also polar. The fatty acid tail is hydrophobic because it is nonpolar and repels water molecules. Cell membranes are phospholipid bilayers, a double-layered structure in which the hydrophilic heads of the phospholipids face outward. The middle of the membrane contains the hydrophobic tails of the phospholipids, an arrangement that shields them from contact with water molecules. Whereas membrane lipids act as a general barrier around the cell, membrane proteins have specific functions. For example, they may act as enzymes, receptors, anchors, or channels. There are two major populations of membrane proteins: integral proteins, which are part of the membrane structure and usually span the width of both phospholipid layers, and peripheral proteins, which are bound to the surfaces of the membrane.
Okay, the cytoskeleton functions as a way of providing the cell shape and support. It provides an internal protein framework that gives the cytoplasm strength and flexibility. And what you have in this particular slide is a list of different cytoskeletal components, some remarks about them, where they're found, and what they do. Okay, so you can see some of them diagrammed out here: microfilaments, intermediate filaments, microtubules, thick and thin filaments, centrioles, cilia. Okay, and what I want you to do is review this material because it's important, and it may appear on a quiz or on a test or in your homework. Now, at some point during the life of most cells, there's going to come a time when the cell has to reproduce. And for body cells, with the exception of the cells that produce the sperm and the egg, this process is called mitosis, which involves the duplication of all the genetic material in the nucleus, the division of that duplicated genetic material into two separate cells, followed by the completion of the division of these two new cells. So from a single cell at fertilization through mitosis, we get approximately 75 trillion cells. Mitosis produces a pair of daughter cells, each with a complete set of 46 chromosomes, while meiosis produces either sperm or egg cells with only half the same number of chromosomes. In a human being, sperm have 23 chromosomes and eggs also have 23. And when they fertilize each other, we restore the normal diploid number of 46. This number here is called the diploid chromosome number, sometimes designated as 2n, and this is the haploid chromosome number, sometimes designated as n, where n is an entire complement of chromosomes. The lifespan of a cell varies from hours to decades. Some cells will self-destruct after a period of time in a process known as apoptosis, or programmed cell death. We can divide the cell cycle into several phases. In interphase, the cell is performing its normal functions in the body. If it's preparing to divide, the chromosomes are duplicated and all the proteins are synthesized. Mitosis is where the genetic material in the nucleus is duplicated and then distributed to two new daughter cells. That's generally followed by cytokinesis, which is the division of cytoplasm to become two separate complete cells. So mitosis always happens first, and that's followed 99% of the time by cytokinesis. There are a few exceptions in the human body where that does not occur, but they are quite rare, and we'll note them when we get to them. In DNA replication, we have to make a duplicate of all the information that's in the nucleus. So the DNA strands will unwind under the power of enzymes. An enzyme called DNA polymerase will bind to the exposed nitrogenous bases and promote bonding between bases on DNA and complementary bases in the new strand. And so what happens as we move down the DNA molecule is that we build a new strand based off information in the old strand. As the DNA polymerase works its way from one strand to the other, wherever it encounters an A in the old strand, it'll put a T in the new strand. Wherever it encounters a C in the old strand, it'll put a G in the new strand, and vice versa. And in this way, we get two complete copies of the information. One copy will go to one new cell, and the other copy will go to the other new cell. At the end of DNA replication, the original strands have completely separated, and two new identical DNA molecules are formed. Centrioles and other enzymes and proteins have been synthesized, and the cell leaves interphase and proceeds to mitosis. So here you can see an example of what the end products of the process look like, and this happens for each of the 23 pairs of chromosomes. In prophase of mitosis, replicated centrioles move to opposite sides of the nucleus. Spindle fibers connect the centrioles. The spindle fiber's purpose is to position the chromosomes in the center of the cell, and then ultimately to pull the duplicated chromosomes apart. Nuclear envelope will disintegrate, and the chromosomes coil tightly enough to be seen under light microscopy. In metaphase, the chromatids align on the central metaphase plate, and in anaphase, the centromeres of each chromatid are pulled apart. The chromatids are pulled along the spindle fiber towards opposite sides of the cell, and then at telophase, the nuclear envelope reforms around each new group of chromosomes. The nuclei enlarge, and the chromosomes uncoil into chromatin, and that is followed generally by the beginning of cytokinesis. In human cells, the formation of a cleavage furrow marks the beginning of this event. So here you can see the different phases of mitosis diagrammed: interphase, prophase, metaphase, and then anaphase, telophase as we approach the opposite ends of the cell, and then cytokinesis, where we divide the cells in two. So take a listen to this quick video on mitosis.
This soccer player's knee will be as good as new in a few weeks, thanks to mitosis, a type of cell division that generates new cells for growth and repair. Let's move into a cell to witness the events of mitosis. Before a cell can divide, it must first duplicate the chromosomes stored in its nucleus. During chromosome duplication, several bubbles open up along the chromosome. Each bubble grows until it merges with an adjacent bubble. Each chromosome now consists of two identical copies called sister chromatids. Getting closer, we see that each sister chromatid consists of DNA wound around small proteins called histones. The sister chromatids begin to coil into tight helical fibers. Outside the nucleus, centrosomes that duplicated earlier move away from each other to opposite sides of the cell. Microtubules extend from the centrosomes, forming the mitotic spindle. Back in the nucleus, the DNA forms loops, becoming more compacted. These structures fold back on themselves, eventually condensing into a shorter and thicker chromosome consisting of two sister chromatids. As the chromosomes continue to condense, the nuclear envelope breaks up. The array of spindle microtubules is now extensive, and the chromosomes are fully condensed. Spindle fibers from each pole attach to protein structures located at the centromere of each sister chromatid. As the chromosomes are bound by spindle fibers from opposite poles, they move first one way and then another. The counteracting forces of the spindle eventually cause all the chromosomes to end up at the center of the cell, as if arranged on an imaginary plate. The sister chromatids are released from each other, each becoming a full-fledged chromosome. They are moved toward opposite poles of the cell, pulled along the spindle fibers attached to them. At the same time, overlapping spindle fibers that are not attached to chromosomes continue to lengthen, pushing the poles farther apart. Once the chromosomes arrive at their destination, they become less condensed. Two new nuclear envelopes form, completing mitosis, the division of one nucleus into two genetically identical daughter nuclei. The cytoplasm divides by the process of cytokinesis, forming two separate daughter cells. In your body, millions of cells divide every second, providing new cells for growth and for repair of damaged cells.
Occasionally, mitosis can proceed in an unregulated fashion. When this happens, irrespective of the needs of the human body, we call this cancer. It's often characterized by changes in the information in the nucleus, producing an abnormal cell. It's most common in tissues that divide rapidly, primarily due to the fact that there's the potential with every mitotic division of making a mistake in a gene that has a role in controlling cell division. And if that happens, there's the possibility of producing cells that divide in an uncontrolled fashion. This produces a tumor mass. The benign tumor mass will remain in one place and not spread throughout the body, while a malignant tumor will divide rapidly and spread. They produce chemicals that stimulate blood vessel growth. This is called angiogenesis, in order to feed the tumor. The tumor grows and the cells migrate to other areas and establish new tumors in a process called metastasis. And the problem, of course, with this is that these new tumors and the original tumor need fuel. They need oxygen and nutrients in order to grow. So what happens in cancer that's uncontrolled and untreatable is that it takes so much oxygen and nutrients out of the blood that healthy tissues can't survive, and the result is that the individual with the disease dies. Malignant cells no longer perform the original function or perform functions in an abnormal way. As an example, a malignant tumor of the thyroid produces abnormal amounts of thyroid hormone. Cancer cells compete with normal cells for space and nutrients, and that's what eventually kills the organism, kills the human being. Generally, these mistakes that happen in cells as a result of cell division are intercepted or controlled by your immune system before they get a chance to spread. But in the event the immune system misses one of these abnormal cells, it has the potential then to grow and metastasize and spread throughout the body. And generally, the systems that they use to facilitate their spread are the circulatory system and the lymphatic system. They're kind of like subway trains to go all around the body. The cell membrane is important because it keeps all the cellular components in one physical space, and in addition, it serves as a semi-permeable barrier so that the cell can let in things it needs in order to continue to grow and release stuff that it doesn't want or that it wants to secrete in order to fulfill its role in the human body. The cell membrane also serves as a way to tell the cell what's going on in the outside world, generally through proteins that are called receptors that are on the cell membrane surface. And it also serves as a way to indicate that the cell belongs to the human body so that the immune system leaves it alone through collections of molecules on the cell surface that are called antigens. Permeability of the cell membrane determines which substances can enter or leave the cytoplasm. If the membrane were freely permeable, anything could pass through this barrier, but in a selectively permeable membrane, Some components can get through and some can't, while in an impermeable membrane, nothing can pass. And so you can see examples of all three of these below. This would be a freely permeable and an impermeable membrane. The true state of the membrane in living cells is a semi-permeable membrane, where some stuff can pass and other stuff cannot. So how is it that the cell allows stuff to penetrate the cell membrane? The permeability of substances is based on their size, their electrical charge, their shape, and their lipid solubility. Materials move in and out of the cell through passive mechanisms that don't require ATP. Examples of this include diffusion and carrier-mediated transport and osmosis, which is the diffusion of water. Active mechanisms need ATP, and these include carrier-mediated transport and vesicular transport. And you can see these diagrammed here in the inset. Okay, so make sure and review this figure in your text. In diffusion, ions and molecules are constantly moving and colliding and bouncing off each other and moving in a variety of different directions. The process of even distribution from this random movement is called diffusion. Uneven distribution produces a concentration gradient. Diffusion restores the even distribution of these substances throughout the medium, whether it be water or a gas. The motion of particles continues, but no net movement occurs. So you can see an example here with a drop of food coloring placed in water. If we wait long enough, eventually the substances in the little purple square here will evenly distribute themselves all across the fluid. And you don't have to put any energy into the system for that to occur. It happens spontaneously, and that's why we call it a passive process. Now, a couple of things affect the rate of diffusion. One thing that affects it is how steep the concentration gradient is from where the substance originates and the rest of the container. The steeper the concentration gradient, that is, the greater the difference in concentration between the substances diffusing and the medium around it, the quicker it'll diffuse. The lower the molecular weight of the substances diffusing, the quicker that it will diffuse, and the higher the temperature, the quicker the substance will diffuse. An ion or molecule can diffuse across a plasma membrane by crossing the lipid portion of the membrane or passing through a membrane channel, like a tunnel or a gated opening. Larger molecules can't diffuse through the membrane. They cross the membrane using a carrier. Individual solutes diffuse along their own concentration gradient and are not affected by the movement of other solutes. The plasma membrane in living cells is freely permeable to water. Diffusion of water is called osmosis. And you can see some of the other substances and how they move across, right? Lipid-soluble substances can freely pass. Other components, including charged particles and small polar molecules, need a special tunnel to pass through, or a pump, and the pumps are often powered by ATP. These channels can either be open all the time, or some channels will have doors on them, so they're called gated channels that will open and close depending on the conditions in the cell. Diffusion is the movement of molecules from a high concentration area to a low concentration area down their concentration gradient. Osmosis is the diffusion of water across a selectively permeable membrane down its concentration gradient from where it's most to least concentrated. Facilitated diffusion uses the concentration gradient as a driving force to move the substance across the cell membrane, but usually the substance is either charged or polar, and so it needs a special gate or tunnel in order to get across the hydrophobic core of the cell membrane. So you can see an example of that in the bottom inset down here. Okay? Movement of materials across the membrane down its concentration gradient assisted by a carrier protein is the definition of facilitated diffusion. Active transport uses ATP in order to drive this movement either with or against the concentration gradient. It is carrier mediated, so there are proteins in the cell membrane that aid in this process, and the substance being moved can either be moved with or against its concentration gradient because we're using energy in order to move the substance in a particular direction. An example is the sodium-potassium exchange pump, which uses one ATP molecule to pump 3 sodium ions out of the cell membrane and into the extracellular environment. For every 2 potassium ions, it moves in the opposite direction. So that's a 1 ATP process. Vesicle formation utilizes the formation of a membrane-bound vesicle in order to either expel a substance out of the cell into the extracellular environment or to move fluids or particles into the cell. So when we move stuff into the cell, it's called endocytosis. When we move stuff out of the cell, it's called exocytosis. Receptor-mediated endocytosis uses a specific molecule that will bind to a membrane protein. Once that binding happens, it triggers the formation of a vesicle, which will then move inside the cell, and that substance will then be deposited in an organelle, or perhaps it'll move across the cell and be expelled from the other side. There's any number of fates for the receptor-mediated endocytosis. Once the vesicle has released its contents, or its contents have been dealt with, the original vesicle can fuse with the plasma membrane again and repeat the entire process. In pinocytosis, a small amount of liquid is brought into the cell by a vesicle that does not have receptors on its surface. Phagocytosis brings solid particles into the cell, and exocytosis moves cellular products or wastes from the inside of the cell to the outside of the cell. So the difference here is just a matter of direction. Okay? In exocytosis, we're throwing things out. In endocytosis, we're bringing things in. And that both processes use ATP because this is not a process that would happen spontaneously. Molecules interact to form cells, which combine to form tissues, which combine to form organs, which combine to form organ systems. Okay, so we can see here the hierarchy of things, and this is one of the reasons why we teach you guys about chemistry, so you can understand what atoms are, where molecules come from. Collections of molecules can become cells, collections of cells can become tissues, and collections of tissues can become organs, and collections of organs can become organ systems, which altogether form the organism. Histology is the study of tissues. There are 4 primary tissue types. These include epithelial, connective, muscle, and nerve. Okay, so epithelial tissue is hallmarked by the fact that when you look at it under a microscope, it's got a lot of cells with very little space between the cells. So the most common type is the type that forms barriers. It covers exposed body surfaces. It lines the GI, respiratory, reproductive, and urinary tract. It surrounds internal cavities and fluid-filled cavities. It lines the inner surfaces of blood vessels. And why is it so good at doing this? Because the cell connections are very tight between this tissue type. As a result, substances that are going to pass through an epithelial layer have to go likely through the cell in order to get from one side of that layer to the other. In addition, because of these tight junctions, it's very good at forming seals and keeping things out. That's why it's great as a covering for the outer surface of the skin and lines a lot of our organs, because a lot of times the things that our organs produce or the things that our organs contain have to be isolated in a specific area in order for that physiological function to occur in the most efficient way. Connective tissue is kind of almost the reverse of epithelial tissue, and it's characterized by very few cells and a whole lot of extracellular substance. Now, sometimes that substance might be semi-solid. Sometimes it might be a liquid. Um, Sometimes it might be a solid. It's diverse in appearance. It contains specialized cells and matrix, that's the stuff between the cells, which can be composed of a variety of things. In this type of connective tissue that we're looking at, which we call loose connective tissue, the matrix is composed of protein and ground substance. It has a varying consistency. In the liquid connective tissues, blood and lymph, the interstitial fluid is the material between the cells, is the matrix. So in blood, that's plasma, and in lymph, that is filtrate. Bone has a solid matrix, which is mostly protein and calcium salts. And as the name implies, what is the role of connective tissue? It's to attach other tissue types to each other. So in a sense, it's the body's glue. It also provides a mode for transport with the liquid connective tissues, and it provides the body a framework, such as the connective tissues, cartilage, and bone. Okay, but again, anytime you look at a connective tissue under a microscope, what you'll find is very few cells and a lot of extracellular substance. And it's the extracellular substance, generally speaking, that fills the job description of that particular tissue. Muscle is unique in that it has the ability to change its shape, produce force by contracting. It moves the skeleton, it supports soft tissue, it maintains blood flow, it moves materials along internal passageways, it stabilizes our body temperature. Now you might say, okay, I can see some of those, but how about this moving materials and body temperature stuff and maintaining blood flow? Well, the blood flow part of muscle tissue is the result of smooth muscle, which is found primarily in the linings of blood vessels and in the linings of organs, in the pupil of your eye, and attached to every hair in your body. And it's a special type of muscle tissue that works without us having to consciously control it. It's governed by the autonomic nervous system. And so one of the ways that we can maintain our blood pressure is by varying the amount of pressure that smooth muscle puts on the blood that flows through our blood vessels. So the tighter these muscles squeeze our blood vessels, the greater the pressure becomes, and the less squeeze we have, the less the pressure becomes. And we call this vasoconstriction and vasodilation. So let's write those terms up here so that we don't lose track. Okay, so vasoconstriction which tends to increase pressure, and vasodilation, which tends to decrease pressure. Okay. It moves materials along internal passageways in a very similar fashion. There's smooth muscle, for instance, in the digestive system that contracts, producing wave-like motions called peristalsis. Let's write that down, peristalsis. And that helps push food from our throat all the way to our anus so that we can absorb the nutrient content along the way and dump the waste products out once we've removed the nutrient content. Stabilizes normal body temperature through, in part, vasoconstriction and vasodilation, in part through the production of goosebumps. Okay, remember goosebumps? Okay. It also has a role in maintaining body temperature by changing the cardiac output, which is simply the amount of blood that the heart pumps out in a certain period of time. Okay. In addition, it engages in shivering when we get cold. which turns chemical energy into heat. So we'll say generates heat. Okay? So all kinds of interesting things muscle tissue can do. And there's three major muscle types: cardiac, skeletal, and smooth. Skeletal muscle is attached to the skeleton and is under voluntary control. Cardiac muscle is only found in the heart and is not under voluntary control. And smooth muscle is found in the linings of blood vessels, within glands, in the linings of organs, and is also not under voluntary control. And another thing that you'll notice about this tissue type is that in skeletal muscle tissue and in smooth muscle tissue, you have these unusual structures that are called striations. They're these little vertical lines that you can see here in the skeletal muscle tissue, these little tiny vertical lines. You can also see them in cardiac muscle tissue. Okay, these are striations. And again, they're only found in cardiac and in smooth, or cardiac and in skeletal muscle tissue. And they represent areas where muscle proteins overlap in order to produce contraction. When we get to the chapter on muscle tissue, we're going to find out that in order to produce force, the muscle tissue has to shorten. And the way that it shortens is that the muscle proteins kind of walk over the top of each other, and that causes the entire muscle cell to shorten, which causes the entire muscle to shorten. And they use ATP as an energy source. Neural tissue is specialized for another task, and that's to send and receive electrical signals. And as a result, it has a lot of interesting extensions that come off of a cell body. You can see here the cell body, or the soma, of the neuron, and then these little projections here, these short ones, are called dendrites, and then you've got these long projections that are called axons. In fact, if we were to draw a sort of a nerve cell model, it would look, let's do this in black, it would look kind of like this. You're getting just a tiny piece of the nerve cell in the little picture there. If I were to draw an entire typical nerve cell, this would be the body. Then you have this long process called the axon. Nucleus would be here. And the way that we send and receive electrical signals is that electrical signals, and let's put electrical signals in, I don't know if yellow will show, but we'll try it. Electrical signals start here. Okay, and then they're going to move from the cell body, from the dendrites to the cell body and down the axon. So let's use blue for direction of travel. Okay, so the electrical signal moves, sorry, we'll use green. Electrical signal moves in this direction and all the way down to the end of the nerve cell, and that causes it to release something called a neurotransmitter, which is a chemical that will induce an electrical change in a target cell. Okay, so that's how electrical information moves through a typical nerve cell called a neuron. There's two types of cells in neural tissue. There are the neurons, which actually send and receive the electrical information, and there are the neuroglia, which support, protect, and isolate the neurons from the rest of the body. So you could think of the neuroglia as the personal assistants to the neuron. There are two anatomical divisions of the nervous system. The central nervous system is made up of the brainstem, and the spinal cord, while the peripheral nervous system is all the other nervous tissue outside the central nervous system. Okay, occasionally tissue gets injured, and if we don't fix the injury, we're going to be prone to infection and disease and death. And so the tissue response to injury results from the release initially of chemicals and the exposure of proteins that normally are kept within the integral structure of the tissue. Okay, that's sort of an SOS saying that I'm hurt. Okay, the result of inflammation is increased blood flow to the area from vessels that dilate near the area, bringing more blood to the area. The increased permeability allows white blood cells that are in the circulatory system to ultimately find the area of the injury or infection, leave the blood supply, and then migrate to the injury and perform their task. Pain is designed to warn the body that we have an injury and not to use the affected area. Okay. The increased blood flow and vessel permeability jack up the body temperature, which tends to kill pathogens and increase the rate of reproduction of white blood cells. It increases oxygen and nutrient delivery to the area so that tissue repair and pathogen fighting can ensue. It increases phagocytosis by bringing white blood cells to the region. It also helps to remove toxins and waste products. And if we don't do that, we create conditions that cause cells to die. Okay? Regeneration occurs after damaged tissue has been stabilized. Fibroblasts make collagen fibers to stabilize the injury site, and this produces a dense collagenous framework called scar tissue in many tissue types. Normally, scar tissue is remodeled to look somewhat normal in appearance. Permanent replacement of normal tissue by scar tissue is called fibrosis. Now, not all tissues will scar when they're injured. Some tissues will regenerate. Examples include things like neural tissue. Okay, neural tissue, liver tissue. Liver tissue will regenerate fully functioning liver cells and liver tissue networks in the event that it is injured. So it has the ability to regenerate with full function, whereas a lot of other body tissues, such as connective tissue, epithelial tissue, when they're injured, they generate this scar tissue, which is a temporary patch between where the injury occurred and the outside world, and this is to prevent further infection. Okay? All right, that brings our third podcast to an end. Again, make sure and review this material. Let me know by email if you have any questions, and I will see you in the next podcast. Thanks for listening.